Give me 1 second.
Hold on.
I'm trying to be calling him all the time. It's not letting me unmute it.
Good morning, everybody. It is our pleasure to present the third CAHFS technical seminar today. And it is especially my pleasure to introduce Dr. Brad Pesavento. Dr. Pesavento is there at CAHFS lab at Davis and has a very impressive work history. And starting with his education, he received his Bachelor of Science at the Department of Chemistry and Biochemistry, UC Los Angeles in 1994, and his PhD in Biochemistry and Molecular Biology, Baylor College of Medicine in Houston, Texas in 2002. For the last 2 years, Dr. Pesavento has been the electron microscopy section manager at CAHFS there at Davis. So for the last 2 years, prior to that, he was business development for ICOR Incorporated in San Francisco for 2 years, strategic development in the Gastric Cancer Foundation in San Francisco, project finance with Gentech Incorporated in South San Francisco for 2 years prior to that, account manager for Biorad Laboratories in Hercules, California for 2 years, research scientist at the University of California at Berkeley for 2 years, adjunct professor, Las Positas College in Livermore from 2003 to 2010, biophysicist, Life Sciences Division, Lawrence Berkeley National Laboratory in Berkeley for 2 years, postdoctoral scientist at Lawrence Livermore, 2002 to 2006, pre-doctoral fellow, Department of Biochemistry and Molecular Biology, and the W.M. Keck Center for Computational Biology, Baylor College of Medicine in Houston from 1996 to 2002, and a biomedical scientist, Lawrence Livermore National Laboratory in Livermore from 94 to 96. I don't know how you keep track of all that, Brad. You've been busy. Yeah, no, that's great. It's our pleasure to have you here today. Very much appreciate taking the time out of your busy schedule. And at this point, I will turn it over to you. And I know one thing we're all dying to hear. It's just about your title, from end to end, electron microscopy at CAHFS. So, Brad, I'll turn it over to you.
Morning, everyone.
Morning.
Happy to be here. Beautiful weather this time of year. So I'm running the electron microscopy section at CAHFS. Most of you are familiar with CAHFS. I'll talk to you a little bit about electron microscopy to go through. I'm going to give a little bit more depth into my background just to help you see how I got to where we are. That's a picture of me with the new microscope, which EDFA was instrumental in helping us get, Dr. Jones especially. But yeah, for the title. So there's two meanings here. CAHFS is a statewide system. We have branch labs from one end of the state to the other. And so San Bernardino to Larry, Turlock, Central Valley, and then Davis. And Davis is a full service branch, which means we have all capabilities, including electron microscopy. And we're one of the few EM centers in the country, the primary one on the West Coast, so we're getting samples from all over. But end to end, because, as you know, animals get sick up and down from mouth to the other end. So we look at viruses from all over, from all parts of animals, and we'll get to that towards the end when I give you some examples of more of these things. So we really do take care of everything up and down the state from all parts of the animal. So I'll go into a little bit about my background, as Kent mentioned, and I'll talk, during that point, I'm going to mix in some primer on microscopy. So just general show of hand, how many people have ever used an electron microscope? How about light microscopes? Okay. So I'll even give a little primer on light microscopy just to catch some of you up who may not have done it since high school or college. I'll talk about how we've been monitoring dynamic CAHFS, and then I'll present some recent cases, some things that are important, and what could be coming. You never know. So my background, so I started off at UCLA in, graduated in 1994. I'm from California. I grew up in the Bay Area. My parents actually are from Sacramento. My grandparents lived on Land Park Drive, so for me, this is sort of my second home. But my bachelor's was in biochemistry, and towards the end of my bachelor's, I worked in the hospital at UCLA in the molecular pathology department, and so that was where I was first exposed to illnesses. This is human pathology, but sort of learned good practices and how to manage cases. From there, I did 2 years after college. You know, as most people try to figure out what they want to do next. I worked for the Human Genome Project, that was a payday, and I was in the mapping and sequencing group, and we did a lot of high throughput and automation. So I was programming robots, which was really cool to get out of college. This was the Orca robot here. It's an arm that handled microtiter plates. So we were manipulating clones, vectors, different DNA sequences, and they were mapping them through sequencing, so they would figure out how to line everything up. This is on chromosome 19 that Livermore had, but it was one of the big centers for genomics before they were all merged and got into the high throughput sequencing. So this was a lot of fun. Did that for a couple of years, and then realized that I wanted to get my PhD. I wanted to stick with biology, biotechnology, and computational efforts. So I went to Baylor College of Medicine in Houston, and this is the Houston Texas Medical Center. Has anyone ever been down there? It's huge. It's basically a city within a city. So that's Houston downtown there, fourth largest city in the country. And this is the Texas Medical Center, and it, on a daily basis, has between 80,000 and 100,000 employees, not including patients. So as you can imagine, traffic and parking are issues. I used to ride a shuttle bus and walk from Rice University because I had a fellowship through Rice. So big campus. There's 12 or 13 hospitals, 3 or 4 medical schools, nursing, dental. Texas A&M had a presence there. And there I studied both conventional and cryo-electron microscopy. And I'll talk a little bit more about what cryo-EM is, a specialization of electron microscopy. And my PhD work was on rotavirus, the structure and function. This is a virus that infects humans and animals, generally infants or babies, and it could be quite deadly. At the time when I was working on the rotavirus, there was no vaccine, and the vaccine was being introduced and it had to be withdrawn, so there was a lot of importance to the research that our lab was doing. And then to aid the cryo-electron microscopy, I did computational biology, which involves handling large data sets to electron micrograph images were digitized and reconstructed in the computer. I'll show a little bit more about that. Following that, I went, I'll back up here with microscopy. So there's different levels at which we can visualize things. Right? We have our eyes so we can see things in the natural world down to a certain size. Once we get down to a certain small size, we need to aid how we look at things. So the microscope was invented. And this just gives you a scale, metric units of things. So obviously blue whale, human, mouse, ant, we can see these things visually. We don't need any assistance aside from glasses. But once you get down below a millimeter, you really need some assistance. So light microscopy becomes very useful. And you can see the cellular layer level and some bacteria. But once you get to around the micron or micrometer level, the light microscope is ineffective. And this is due to the wavelength of light. So visible microscopy wavelength, you can see things between around 400 and 700 nanometers. Electron microscopy, however, can go down into the angstrom resolution. So people in material science can actually look at atoms. Now, in biology, we don't need to go down that far, but the microscope has that power, that capability. And so between light microscopy and electron microscopy, we can look at the cellular, bacterial, the virus levels of life. And just to give you that tutorial in light microscopy, I promise, you have several important components. You have your illuminator, which is your light source. This is generally located at the bottom of your microscope. You have a condenser, which does what it says. It condenses the light, or it collimates it. It brings it together into a more column shape. So instead of like a cone or a diffuse beam, you can get a very sharp beam column of light. And what you have is the stage, which is where you put your specimen. And this can move your area, your slide or your object of interest up and down, back and forth. So you can actually go in the X direction or the Y direction. And you've got your focus knobs, which obviously helps you to sharpen that image. And on a light microscope, your focus, especially your coarse focus, will raise or lower the stage. So we call that the Z direction. You've got your objective lenses, which those of you who use light microscopes know you can rotate that into different powers or magnification. Generally, they range from 4x to around 100x. When you go up to over 100x, you may have to use foil because of the refractive index of light. So that's on the revolving nose piece. You've got the head of the microscope. So this is a season top head, which is where you can rotate the eyepieces so you don't have to change the focus. So your interpupillary distance, everyone's got a little bit different separation between their eyes. So that helps to adjust that. And then your eyepiece, which we call an electron microscope, these are called binoculars, but they help you to zoom in on something which is down here. So you actually need to magnify it a little bit further. And so you can adjust the focus on those. So that's how we look at the cells, some bacteria. And it's, compared to an electron microscope, it's not very complex, but it does take some training to get used to how to operate. Now, as I mentioned, light microscope is useful between 400 to 700 nanometers. That's the highest or the smallest object you can see without going to additional technology. Image is differential color absorption, so you can see colors in the light microscope. Uses glass lenses. 200 is really the absolute limit of something. You probably won't be able to see that. Samples in histology tend to be embedded in paraffin. So a tissue from an animal will be put into a paraffin block and then sliced off and put on a glass slide. Those specimens tend to be 5,000 nanometers or 5 microns in thickness. And a really good light microscope may cost you $20,000. On the other hand, when you want to go beyond that resolution, you need to go to electron microscope. So you can get up to 0.04 nanometers at an 80 kilovolt acceleration. I'll talk a little bit about the voltage. Image is formed by scattering of electrons. So we're no longer in the light realm because the wavelength of light is not small enough to see those objects. The objects are smaller than that wavelength of light. The lenses are made out of electromagnets. So I adjust the focus and the different components of the electron microscope by changing currents in these magnets. I'll show a diagram in a second. In biology, 2 nanometers is a reasonable resolution. With cryo-EM, you can go higher because you're adding together multiple images. Samples for thin sectioning are commonly embedded in resin. Epoxy is sort of a plastic, it's yellowish in color. It looks like amber. That's the origin of that. And specimens tend to be between 60 and 100 nanometers in thickness. Now, a virus can be 100 nanometers, so you may actually cut through a virus if you cut it too thin. And the cost, this is sort of your starting cost. So our scope was in this range, but the higher voltage microscopes can go up into the millions of dollars. And of course, those become shared instruments. You can't have one to yourself. So I'm fortunate in that we have the scope and I'm the primary user, and I'm very grateful for that. So here's a diagram of what a TEM or transmission electron microscope looks like. So there's another type of electron microscope called an SEM, scanning electron microscope. That does not penetrate through the object, so the resolution is not as high. You get more of a surface. You may have seen some of these pictures that look three-dimensional of ants or insects. These are using SEM. TEM is at a higher resolution, and it requires a much more powerful system. So as opposed to a light microscope, the source of illumination comes from the electron gun, comes from the top. So everything is inverted now. So the illumination comes through the electron gun at high voltage. The TEM that we just got in CAHFS can go up to 120,000 electron volts. We generally operate at 80,000 electron volts just to preserve the sample, because at that high voltage you will melt, it will burn through your sample almost like a laser. So I turn down the voltage and I'm generally looking at things. We have our condenser lenses. These are the electromagnets. This is a cross section. So whenever you see these X's here, it's actually a ring like a donut that's been cut in half. So you're seeing the two halves of the ring there with the X. So these are circular rings in the cylinder. So you have your condenser lens and your condenser aperture. So that will concentrate the electron beam. As electrons are spraying off from this gun, there's actually something at the tip here called the filament, which is heated up when you turn on the current. And it's spraying electrons sort of like a fire hose. And if you've ever seen the hose, water can go in all directions, but if you turn the nozzle, you can focus it. So the condenser does the same thing. We turn it up, and we exclude any electrons that are not within that range, that diameter that we want. And then we have our specimen, which is inserted from the side, and it's right there. And so the electron beam passes through the specimen, and then it hits the objective. So the objective and the specimen are right next to each other. The specimen in an electron microscope is, you could compare it to that stage in the light microscope. It's the plane where you put your slide. And in your objective lens, you can change your magnification there. And then what we have are different lenses, intermediate and projector lenses, to magnify that image. So the image is formed from your specimen, and then we amplify that to be able to see it. And that hits the fluorescent screen at the bottom. So this is a green screen that we use. And then you can look actually through the binoculars at your screen to see your image. But nowadays, we don't use these. We have a camera down here. We have a digital camera. So everything's on the computer, so you don't have to bend over and turn the knobs. You can just sit there, and multiple people can share the microscope. Okay, so that's sort of your primer. So back to my background. So I studied the rotavirus structure. So these are the proteins and the corresponding RNA segments to rotavirus. Rotavirus is a double-stranded RNA virus. It has 3 capsid layers. This is the outer layer called the triple layer particle. Then the blue is the double layer particle, and then the inside is the single layer particle. And the triple layer particle is the full infectious form of the virus. And this is artificially colored because we don't have color at that level. But this virus is around 80 or 90 nanometers in diameter if you include the spikes. The spikes are what are attached to the cell receptors during the infection. And this virus enters fecal-oral route. Okay, so it gets ingested, and at pH 2 it becomes activated. So it's actually very sturdy. It can withstand very acidic environments. So that's why it's activated in your gut. Those spikes become ordered at pH 2. They then attach to the cell. The outer layer is removed, and the inner layer is transcriptionally active. So the RNA comes out and makes millions of copies of virus, and you get sick. This results in gastroenteritis, dehydration, vomiting, diarrhea. It can be deadly. So that's in a nutshell how rotavirus works, and that's typical for a lot of the viruses that we see at CAHFS or enteric viruses causing these things. Now, to get even more technical, since it's a technical talk, and this will probably be, this in the computer will be the last bit of technical information. Viruses, a lot of viruses have an icosahedral architecture to them. So their structure is like a 20-sided dice, and you can turn it different angles and look at the different symmetry. So you have the 5-fold symmetry here, so you see this pentagon shape from the top. And if you follow this yellow triangle and rotate it around, you get the 3-fold. So that's your triangle in the middle, you have 3 sides. And then if you turn this to the left a little bit along this edge, then you have a 2-fold symmetry. And using those symmetry operations in the computer, you can find all the different orientations in a single image and reconstruct it three-dimensionally. And you can actually break this down into a smaller unit. But basically, you take a picture, you scan it in the computer program knows how to orient the virus, and then it's going to take all those different orientations and merge them in the computer and create a three-dimensional image. And that's how this is done. So this is your two-dimensional image of the rotavirus. You can actually see some of the spikes coming off. So this is an EM image. This is in ice. So it's been frozen, flash frozen. So it's vitreous ice. So that ice has no crystallinity to it. So it doesn't affect any of your image process. It makes it clear like a glass, like a sheet of glass. So these particles are embedded in that ice. And what the program does is it takes a box, draws a box around each of them, creates a mask where it excludes all the external area. And it does something called a Fourier transform, which I won't show you the formula for, but it actually interprets which is the upward portion, what's pointing up, and it's probably right here is your 5-fold vertices. So this could lie anywhere on that plane. This is what the Fourier transform looks like. So it's not very informative. But when you have enough of those particles, maybe a couple hundred, you can put them back together and make their structure. And now, Tuan, if you could click on the middle of that, this is a video showing the PH7 virus. So after the spikes have been activated, it goes PH7 and the PH2, they become activated. And what we did was we actually were taking out of the ordinary and we treated with PH11. And that blunted the spikes. And so it was no longer infectious. But it was still recognizable by antibodies. So that helped a lot of the folks at Merck that were designing the vaccine for the virus. So that's the spike after it's been stunted. It's no longer infectious, but it's still recognizable by the immune system. And so you can see as it's rotated through, you can see the different vertices. And these are the channels where amino acids or nucleic acids could go in or out of the particle. Now this video, that's actually pretty old. When I first made that video, computer, it would crash all the time. So it's pretty impressive how computers have come along. Okay. This is also a video. This is a cartoon illustrating that the RNA in the inside of the rotavirus can condense when you change the pH. And so if you click on that one also, just click on the middle of it. And so when we take it to the high pH, pH11, the RNAs will condense. And that was actually seen in the image on the electron microscope. You can see this dense core. And it was no longer able to transcribe the RNA. So that was a very important finding. So we published that and it was put on the cover of the PNAS. And that helped me to graduate. So after that, I went to Livermore Lab, and this is after 9/11, so there was a lot of interest in Homeland Security. So I came back to Livermore, I knew folks there, and so I worked on several projects there, advanced electron microscopes, biodefense, continued with the computational biology, and I actually had a collaboration at UC Davis. So at Livermore, they had an electron microscope. This is a Philips 300 kilo electron volt scope. And we were upgrading that one to look at infectious agents. This scope is primarily used for plutonium. As many of you may know, Livermore is a nuclear weapons lab. And so this was at that point when I started to put in some of our select agents. It was considered the dirtiest microscope in the world. Just getting into that room was no fun. But those computer images I showed you, we were able to use the supercomputers at Livermore. So what would take me a week before, it took me about 2 minutes to generate structures. So that was the interest in identifying viruses there. I also worked with Cheng's group at UC Davis. This is an example of a hepatitis E virus, maybe a little hard to see, but these were smaller particles, 27 nanometers diameter. And this is a 50,000 fold magnification, and this is a 200,000 electron volt microscope. So the higher voltages you go, the better your images will be, but the more danger you're introducing of frying your sample. And since these were icy with melt holes as you took the pictures, so you had to shoot first and ask questions later. I also worked on a project called the NanoDisc project. So these were little membrane discs that we would stick different proteins in to create antigens for effective threat counter agents, as we say. These were very small. These were 8 to 10 nanometer diameter particles, and they tended to stack. It was very interesting. They formed these long stacks. This is a 60,000 magnification image. I also worked on a very advanced microscope at Livermore called the dynamic TEM. This is a microscope that had 2 lasers that came in from the side. So there was a separate room on the other side of this wall. Livermore is known for having lots of lasers as well as nuclear elements. And so these lasers would come through and activate the cathode, which is the source of electrons, and it would pulse it. And so we were able to do time-lapse images and make movies on the electron microscope. So this is sort of a very R&D, very cutting-edge microscope. And they actually had a sample that could hold liquids, so we could put in bacteria and watch them swim around as the laser was giving and make movies. And that resulted in an award from R&D. So this is an invention, basically, with the company that manufactures the microscope. So this is a very cutting-edge microscope. It's now being produced for certain materials testing. A lot of people in electron microscopy work in Silicon Valley for materials. I worked on some projects at Berkeley with batteries. So there's a lot of interest from the non-biological world in pushing the frontiers in electron microscopy. So after Livermore, I went to LBL. I worked on some bioenergy projects, some advanced EM. They have a lot of very impressive microscopes at the National Center for Electron Microscopy up on the hill. So it was a very nice place to work. It's a very competitive place to work, but very, very cutting-edge equipment. So this is a molecular foundry, and the National Center for EM is the building next to it. They have a Zeiss Libra microscope. This is monochromated, so all the electrons are the exact same wavelength. So that's very useful to get a very clear image. They have the FEI Titan. This is called the soft team. So this is aberration corrected microscopy. So the lens is corrected for any skewing that you would have in your image processing. This is about 1.5 stories tall. And then the team, which is 2 stories tall, is the super high resolution microscopes that could go up to 1 million electron volts for looking at atoms and materials. Generally, well over half the money invested in microscopy now is for batteries because of the need for energy efficiency. You know, if everyone with solar panels could save their energy, they could use it at night, and you wouldn't have to, you know, take PG&E or SMUD anymore for electricity. So that's, and Tesla, of course, batteries for cars. So that's really a huge investment. The rest of it is from semiconductors, Silicon Valley companies. But biology is still important. So we have some groups at LBL. So I was using the tomographic cryo-EM where the specimen can rotate, and we were putting microtubules in these little vesicles, and we call this like a ship in a bottle. So you put the parts in first, and you add the right reagents, and it would polymerize, and we would get different shapes, like this one we called the lemon. And that was just to sort of simulate a cell skeleton, cytoskeleton, so we could understand how our cells develop and what's the breaking point for the membrane, so what kind of forces were involved. So at this point, I've worked after this for Genentech and Bio-Rad and some foundations. And CAHFS needed some help with their electron microscopy. They had an older scope and they wanted to sort of rebuild their program. So they convinced me to come back here because, like I said, I have family in the area, and my cousin actually works at UC Davis, so it was actually sort of coming home. My uncle was a professor at UC Davis, and it's a little more relaxed here than in the Bay Area. I have kids now; it's a better place to have a family, I felt. So they convinced me to come to Davis. So I was brought on board to modernize CAHFS. So this is where we changed pace here to talk about what's going on with CAHFS. So the old scope at CAHFS was no longer manufactured by Zeiss. Zeiss got out of the TEM business, and it tended to have some issues every so often. It would shut off. It started happening more and more often, and that became quite frustrating to help all the pathologists up and down the state that were having outbreaks, and they don't know why. And EM is critical for pathologists because there are emerging strains all the time. There's variation going on, viruses mutate. There's things that pop up, SARS and avian influenza outbreaks that there are no primers for in PCR. There's no antibodies for in immunology. So you know the animals are sick. Or you want to identify where the lesions are. So it's critical for CAHFS to get this resource running. So I know it took several years. I know Dr. Jones was instrumental and Homeland Security was involved. And so when they told me that it was coming, I was, very excited. It took a little while to come. But while that scope finally died, while I was there waiting for the new scope, we used several other older scopes on campus. We used the Philips CM120, JUL 1230, and then they have a new scope in the medical school called the Talus, which is a big box. It's a little bit challenging to operate when you don't have any buttons on it. It was completely computer controlled, but they have a lot of users and they don't want people touching all the buttons. So it was very helpful to have those available. But as I showed you, and so we couldn't afford to buy the biggest scope made by JUL, the 1 megavolt, 3-story tall scope. But this is where you want to really look at atoms and see crystal structures and so forth. So this is in Germany. JUL is a Japanese company, and that's who we ended up buying our scope from. But we did have enough budget that we didn't have to go down this journey. So I thought about getting that for my kids. I don't think it's technically an electron microscope. I think it uses electricity, but it's a light microscope that can talk to you, so it's good for kids to learn. But what we ended up with was the 1400 plus. So this is JOL scope. It's got a large digital camera in here, 4K, 4,000 by 4,000 resolution camera. It can run up at 120,000 volts. It's got a lab 6 filament. This is a crystal. So instead of the older tungsten filaments, which are a wire, like the old light bulbs where the wire would burn out, you'd have to replace it. This is a crystal which will last much longer. And shown here, this is a close-up of that crystal. So it's purple in color, reddish in color, but it's got a nice flat conical shape to it so the electrons come off more evenly. Before with those wires, you'd have to constantly adjust it to get the beam pointing down. We also got a nice 4 specimen holder because I tend to look at 10 to 12 different samples a day. And so putting them in 12 times takes wear and tear on the vacuum system. And here I can just do 2 or 3 exchanges and I can go a lot faster. I can go through 10 or 12 samples in an hour or 2. So that's the rapid results part. So if somebody needs an answer like that, we can get to it. It's generally a mix of samples that are lower priority, like what was this? Was there any virus there? To we need to rule this out because we're going to make a phone call up here because in a few minutes if we don't know. We also, in that budget request, we actually had a little bit of money left over, so we got a new ultra microtome, which is really nice. The old one was way beyond service, and we couldn't get parts to it, couldn't maintain it anymore, so that was frustrating. So we've modernized the lab with new equipment. This is me and Rosa. I've been training her on how to operate the scope. And this is me with Bob Munn, who's a retired microscopist from Med Center at Davis. And Leslie was my supervisor, the pathologist, who helped us, who was instrumental in doing that. So it's in operation. This was brought on board and certified last June. It's been about a year now. And so I spend most of my time between preparing samples and talking to pathologists with virus hunting, so figuring out what's going on. So, you know, a lot of poultry, some horse, cattle, but different species. And we also get samples from researchers for publication. So we have the balance of diagnostic, what is it, and let's get some nice pretty pictures that we can publish and show what we found. So generally for viruses, there's several ways that we try to look at them. The size is an easy one. How big is it? What's the diameter? Is it 20 nanometers, 40 nanometers, 60, 100, 120, 200? The shape. So I showed you earlier that icosahedron. That's the ideal shape that nature has got to balance the surface area to the volume. But you have other shapes. You have helical shapes, and you have amorphous shapes. You also have membrane bound, which can make amorphous on the outside, like influenza and corona. So that affects the appearance. So what do you see in the microscope can help you, and then the experience of the microscopist. So the more viruses you look at, the better you get at picking them out and knowing when something is out of the ordinary. And so some of the common food animal viruses we look at: rotavirus, reovirus, adenovirus, corona, and herpes. I see these probably 90% of the time. We get some pox viruses and other interesting viruses come along. So I mentioned the shape. So the icosahedral symmetry, helical symmetry, complex or uncertain symmetry. So you can have viruses that just don't fit in with what you see. And then the envelope would be a lipid bilayer membrane around the outside, which can give it an amorphous shape. You can also have the non-envelope where you just have that icosahedron or that helix, like a nice rod. Those are very pretty pictures. Viruses can have different genomes. It can be either DNA viruses or RNA viruses. So the DNA ones tend to replicate as a nucleus, and the RNA out in the cytoplasm. They can have a single-stranded genome, so just one half of the RNA, or double-stranded. They can be a linear genome or circular. Earlier in that rotavirus movie, I showed you those coiled like springs. Those were linear segments that were coiled up. But you can have just a big circle that connects to the cell in a big loop. Okay, so at CAHFS, we tend to do diagnostic, those rapid turnaround, and then the thin section EM for the publication purposes. So for direct EM, I'll generally get in the morning tissue preps. So from those branch labs up and down the state, they'll put it in the courier in the morning. You know, at the end of the day, yesterday, they would have put it in dry ice for shipping up to CAHFS status. And I'll also get cell culture from our avian and mammalian virology groups, cell culture. And then thin section EM, this would be like formalin fixed or paraffin embedded blocks, wax blocks. And these come from both our CAHFS pathologists, residents at the vet school, or outside clients. And this is sort of the area where we're growing, is because this facility is unique and there's not a lot, especially on the West Coast. We'll get samples. I recently got some from Canada. I get them from SeaWorld down in San Diego. I got some from up in Portland, Seattle. So as the word gets out there, more and more samples are coming in. And so I'll show some examples here. So this is a coronavirus from turkey intestinal pool. So oftentimes, the pathologist will be working with a group of turkeys that get sick. So they'll pool those intestines, and then if there's a positive, then they can go back to those individual animals to find out which ones are sick. But this was an interesting finding here. So here's the coronavirus. It's sort of an amorphous but rounded shape, and it's got these spikes on the outside, the crown. So corona, crown. This is a rotavirus from turkey and so I've looked at so many rotaviruses. When I see those, I know what I'm dealing with, and Usually those are, you know, the younger birds. They tend to be dying off very quickly. I recently had a case of a bird liver, and it had reovirus. So these are the reovirus. They have a real shape. And this particular sample, so this is a liver that we processed. This was widespread. I mean, this I called it loaded. So it was everywhere, right? So this bird was obviously very sick. But when we looked closer, so this is the lower mag image. That's our scale bar, so 2 microns. This is 200 nanometers. When we look closer in the background, and I don't know if you can see that in this picture, but there was another virus in there. And so there's actually these circoviruses, which are 14 to 20 nanometers in there. And those Rio and circo tend to go together oftentimes in the liver. So this was sort of a 2-for-1 deal. So you always have to keep your mind open. It's not just one virus. It could be multiple. You could have 3 or 4 sometimes. The animals are really sick, and you don't know which one caused which. Is it opportunistic infection, or was it co-infection? Adenovirus, so this is avian cell culture. So again, a lot of eggs are being infected, and we're trying to figure out what is the cause. Adenovirus is a very pretty virus. It's got a nice icosahedral symmetry. You can see the little protein spikes. It's about a 99 or 100 nanometer diameter virus. And with adeno, it's stained. So we use this heavy atom stain to create a contrast. That's why things are black and white, but you're below the level of color. Adenos tend to stain white, and they really pop out. You can see them from a very low mag, like a 500x or 1000x in the electron microscope. So when you get a good adenovirus, you can ID that very quickly. But I like to use this to test the power of the microscope. So here I went up to like 500,000 mag, just to see how good the new scope and camera were working. And so we've got 200,000. Sorry. We got a pretty good image of, you can see the individual spikes. Now, when we did the cryo, we didn't go to this high magnification because we were averaging them together, but here's just a single image to show the quality of the scope. And I mentioned the diagnostic importance for the state. Rapid diagnostics. So we could determine something in 20 minutes. Right? So in an extreme crisis case, which actually I had one a few weeks ago, we didn't know if we had an FAD, if we had a AI outbreak or not. So I was given the trachea of the chicken. And rather than put it through all the normal steps, the processing steps, I just took a stick and I scraped the inside of the trachea and put it right on the EM grid. And I looked around for a while and we found herpes. So we were able to say that it was an ILT, infectious laryngeal trachea, but not an FAD. So this was helpful in ruling something out, so you don't have to make that call, but it was a very rapid identification. So that's the power of EM, especially when something is shifted or mutated, and you're not going to be able to pick it up with your PCR. The other type of EM that we do are those thin sections. So we embed those formalin-fixed tissues and use that microtome. And this is a protozoal cyst vacuole in a bird spleen. So these are the little cysts of the, I believe this was he thought it was a malaria, possibly. So these are much prettier pictures. You have the lesion, you have the tissue surrounding. So this is what we call the publication quality. So you have a lot more information here. You have the blood cells and the surrounding tissue. This is a case of proteinuria in the chicken lung. This is from a PATH resident at one of our branch labs. And what happens here is these lamellae form, these surfactant layers circle around, and this results in something called Merrick's disease. And so this is publication. Actually, this person's coming tomorrow to look at another animal for writing up the paper. And then I also work with the vet school PATH residents, so they're getting exposed to EM. I'm letting them get a little bit of hands-on so that they will, when they have important cases or publications, they will come So this is a bird. This is the gular pouch, so it's a hornbill. So the gular pouch is that thing that it inflates. And there was dark pigment and non-pigmented cells. So where the skin was black, it was these little pigmented structures. And when those are absent, so they call this foamy macrophage. You can see that it's white, so the skin is white in those regions. So at the EM level, you can see the cause of why something's pigmented versus not. And they were trying to determine if this was a disease, caused by disease or not, or it was non-pigmented. And then the last image I'm going to show you, this is something from the outside. This is chromatophoroma. So again, a darker patch of skin with malignant neoplasia on a tegu lizard. And this one, I believe, came from a vet nearby, actually, in this area. And you can see these melanosomes, they're called, that create this darker area in the skin of this lizard. So I have to thank all the folks, you know, in my past work that helped me to get to where I am, both at Baylor, Livermore, worked with folks at UC Davis, and then at LBL. And the folks at CAHFS, so Rich Breitmeyer, who was a previous director, he worked several years to get this scope. He worked here with folks at CDFA and the OES. And then my boss, Leslie Beata, Mandy, our admin, and Kathy Hill, and of course, our new director, Pam Hollinger. So thanks a million for the scope, which is around the order of what a scope like that costs. And then my lab, so Leslie, our supervisor, Rosa, my lab tech, and I have several students that help me with the sample prep. So I think that's it for my talk. I'll be happy to take any questions now, and I know you guys have a process.
We don't currently have any questions online.
Okay.
So we'll open up to the phone.
Okay.
Does anyone have any questions on the phone? No? Okay, we'll move them and we'll take questions from the classroom.
Go ahead.
Did you find anything interesting from SeaWorld?
Yeah, I've gotten several cases from SeaWorld. Yeah.
Anything interesting? Any prognosis?
So the cases that they've sent me have been the thin section type. So I'm currently working on a puffin, skin from a puffin. It's a cute little bird. So he has or she has some kind of bacterial infection. So we're looking at the hair follicle to determine, you know, what kind of bacteria there are. And my background was more virus than microbiology, but bacteriology. So that was a case. And they recently sent another one. It was a bull shark's heart. And we're not really sure what happened to that shark's heart, but you know, cardiology for the shark. So that's an interesting case. But in terms of infectious agents, that was the one, the bacteria.
Yeah, my question is regarding like the sample preparation. Will you be able to run any sample like the swab in EM, or you have to issue the sample?
So the swab, you mean like a throat culture, or? Yeah, we get those. We need enough material to actually see it. So there's a range that we can have too much and we dilute that in. But if we don't have enough, it's really hard to concentrate that when it's a tiny amount. So, you know, I've gotten nasal swabs from dogs. Oftentimes they're looking for something called ciliary dyskinesia for the dogs, and human cases too, where they can't clear their throats, and so they have respiratory problems. And so they'll send a swab from a dog, and they just take a Q-tip and they put it in the nose, and then they drop it in a bottle, and there's not much to work with there, right? But if you do a scraping, you know, use a small volume of liquid, 10, 20 microliters is good, because the amount of sample that we put on an EM grid generally is no more than 5 microliters, and then you dry it, and it forms very dry layers. So a large amount is not necessary. It's more of the concentration that's important. So that chicken trachea, I did a scraping with a stick, and I took that and I put it in a tiny amount of the stain, the heavy atom stain, and then I put that on the grid. And, you know, I was crossing my fingers, but I was hoping that there was enough, because you need enough of an infection to be able to see it. But if the animal's sick, it's going to have enough to find it. So it's a balancing act.
Do you think pulling samples with the text is wrong?
It can. I mean, it needs to be fresh. For the enteric stuff, you know, it needs to be kept cold, and it needs to be processed within 1 or 2 days the most, because the virus will start to get broken down. The bacteria will start to take over, and the tissue won't be fresh. So, you know, if you have something, you know somebody, dry ice, FedEx, get it up there, you know, sooner rather than later.
Actually, I mean pooling samples.
Oh, pooling. Yeah. That's what we get a lot from Tulare Tree-Lock. They're using pools, because that's a more efficient way to find out, you know, if this chicken house has an outbreak. Usually each one of your 50 birds, and so they can pool 5 to 10 birds, and then which of those pools do we see the agent, and then they can go to those 5 specimens that they stay, find out if it was just one animal or all of them.
Examples from clinicians or practitioners that have kind of, you know, that are from a live animal that maybe have gone through the diagnostic process and haven't come up with anything?
I get quite a few. There's different ways to categorize that. You can say there's a Hail Mary path, like trying to explain what's going on. There's a lot of ruling out. You know, rule out neoplasia or cancer or something. Like I mentioned, other tests don't always work. You know, PCR is really good. It's fast. But if something has changed, and you've got the lesion, you've got the infection, but you don't know what it is. And that's, you know, with the herpes, a lot of rotaviruses, there's no ELISAs still that are that good. And any outbreak that's going to happen, we might not be ready, you know, not every diagnostic will be ready to catch it. So this is like the last line of defense. You can take a picture. Whatever it is, you can take a picture. If you have enough material, you can, you know, put it in there, spend some time and find out, you know, what you're doing. So yeah, we do get a lot of mysteries. Yeah.
So if you, I can't remember if it was coronavirus or what it was last year, you said they had a bunch of goats that went to the show.
Oh, the fair, the state fair.
Yeah.
The goats were next to each other, and they were. We got that. I found what looked like coronavirus, but it didn't have the spikes on the outside. And the PCR, I don't think it was turning up, and I think they took it to Iowa. I don't remember what happened on that case, but I remember getting that one. And it was like these goats from different farmers, and they were all sitting next to each other at the state fair, and it was like a big mystery. Yeah. Did they find corona? I looked at it over and over again, and I found like what looked like corona, but without the spikes, so you couldn't make a definitive diagnostic like EM without those, because it could have just been a blob of liquid.
That was my question, like what would the next step be if you found something, say it looks like corona, would then you send it to someone to do genomics, or? Yeah, so when we first got the scope put in, there was an equine outbreak of herpes. But they didn't know it was herpes, so they gave it to me. And I don't know if you guys know Beata, but Beata runs our biotech. Okay, so she came by and I showed her the picture on the screen. And she said, oh, that looks like herpes too. And then they went off and designed something and they confirmed that. So they weren't able to identify it before. So immediately, the day after like the scope was running, we were able to prove the value of the scope by showing that picture, and we were able to identify which strain it was, and then go off and improve the diagnostics. So, you know, it works hand in hand. Yeah. So, $1 million is a lot, and I know, depending on what frame of mind.
Yeah.
But when you speak to a layperson and they hear you spend $1 million, I'm just wondering how you, if you get questions about that, or how you explain it significance. We all give us a slideshow, we all know the significance of it, but how do you communicate that to someone who may not be involved in science?
That's a good question. So, yeah, So relatively speaking, to the average person, it's a lot of money. To big agencies, maybe that's a normal amount of money. But it's important, right? And that's the food safety. So would you rather be safe or sorry? You know, would you rather have an outbreak happen and not catch it, not identify it, and people get sick and the economy collapse, right? So I get a lot of tours in my lab because they like to see it. And so it's interesting. Like kids get it. Like when they come through, it's easy to explain to them. You don't want to get sick, right? I get different groups of people, and they're more interested in technology, but it really is, it's a safety issue, right? So our safety depends on finding these things, and it's actually not a lot of money when you think about how many people there are in the state that we're trying to do that. So, yeah, and it includes, that money includes the service contract, right? So it came with a warranty and everything. So it's like, it's covered for 5 years. Anything that happens, I'm on the phone, they come out the next day. So I'm on the phone a lot with engineers. I can troubleshoot it on my end. But whenever we have any small problems, we do that. That was a problem issue, of course, with the older scope, because they don't make it anymore. The engineers haven't worked on it. And I had the guy there for a week or so, calling Germany every day, trying to figure out what to do, and it was not acceptable. You know, it was just not a way to run.
So what would you do exactly that? You always want the newest and the greatest, but only this is
so this scope will probably be good for 20 years. And when I first did my PhD training, I was using JOL. And there's two main manufacturers. There were several more, but you know, Zeiss is out. Hitachi mainly does scopes for semiconductor. But FEI, which used to be called Philips, they got acquired by Thermo Fisher, so they're a conglomerate, like the Microsoft of bio. And then JOL, the Japanese one. JOL is preferred for its reliability and for customizability. So that scope, we could add on some things and do the cryo that I talked about, or we could do tomography, or we could put in a light scope and do correlated imaging. FEI is moved the other way to be user-friendly. That big box I showed you, we call it a washer dryer. You can't get in there without having the engineer opening up the case. I mean, yeah, I could unscrew it and get in there, but you avoid the warranty. You can't really manipulate it like that. They're going that whole way that software is going, where they want you to subscribe to service contract forever. JOL is depending on the users to have enough know-how to work on it. But, you know, I don't foresee JOL going down because there's so much interest from the semiconductor and materials world that keeps EM going. Cryo-EM just got the Nobel Prize last year. So some of the professors that I knew, 3 of them got awarded the Nobel Prize. So there's a lot of investment going into that component. So there's always new trainees coming out. And that's the other half of the equation. It's not just equipment, people know how to use it. So you want somebody competent with the right kind of background. And that's part of my future goal, is to train people to come in and use it. You know, qualify people, not just anyone off the street, but to have people know how to operate it and interpret their things. Yeah.
How much does it cost to send a sample in just specifically for you?
So CAHFS were subsidized, so we have sort of a reasonable rate. I was looking at some other places, and I think maybe our prices are a little low, but we actually, that encourages more submission. So for direct EM, the quick diagnostic one, from the inside the system, if there's a cap that pathologists have, they can order testing, so that's covered by the cap. But from somebody like at UC Davis, from within UC, they get a discount too. So I think it's between $50 and $100 per sample. For the thin section, that takes a lot more labor and processing, so that's between $200 and $250. But I recently had somebody come here from Australia, and she said in Sydney, their electron microscope person wanted $5,000 to do it. I don't know if that's Australian dollars, but I was looking at University of Florida, and they charge like $1,000. So we're much more affordable. Of course, we're state funded, so that helps. But it's all about providing access. So these important samples can come here, so we can help them out. You know? So it's thousands we don't want to go too low and then be, running in the head all the time. Right? So we're bringing in revenue to the system. But you go too high and people don't understand.
Okay. Thank you
May I
very
ask
much.
one more question?
Yeah, sure.
The Pesavento laboratory has an electronic microscope.
Right.
And where is it?
Peter Woolcock, was he the guy who ran it? I know he's retired, but that scope, apparently that lab was shut down, and that scope was left there. And sort of,
Why the, an antique?
yeah, it was an older, I mean, that was before my time. I think it was 10 or 15 years. I don't know when that lab was shut down, but it was just left there to rust. And I'm guessing that it was scrapped. I don't really know the story.
What a waste, isn't it? Or maybe.
If it was beyond its service life, I mean, it's like a car. Like you were asking, how long will it last? Yeah, 20 years. When it gets to like 30 years old, you can't get parts for that anymore. So you do have to replace it.
Thank you
Okay.
for coming. Thank
Thanks,
you everyone
everyone.
for coming in. David?